so we're going to talk a little bit about structures in microbes, or in a little bit more detail. So moving from microscopy to actual structures. All right, so this is taken out of your book, right? And one of the things we're going to kind of touch on, sort of in the microbial world, is what is the distinction of a cell versus, really any microbe. And the reason I bring that up is because we do talk about viruses, and we're going to talk about virology in the context of microbiology. Largely that's historical because viruses and bacteria were kind of lumped together for a long time because they were traditionally studied as pathogens. But there's some key distinctions, right? There's key differences. And I won't say that the lines won't blur, because there certainly are, as we'll talk about with evolution, intracellular pathogens or intracellular organisms that really cannot replicate on their own. But there are sort of these key tenets of what make up cells, right? So if you're a cell, you have a defined structure. That structure repeats, right? Your structure is something you're going to pass on to your offspring, barring minor evolutionary changes. But then again, evolution, right, is also sort of one of those key components. So you have heritable information that you can pass on, but that doesn't mean that it's a static state, right? So that heritable information can and will change over time, and that does, confer those properties and result in changes across organisms. Cells grow, and also cells have metabolism, right? So they have the capacity to make more building blocks of themselves, right? These are all sort of inherent with, these are all sort of interconnected, right? You have a structure, that structure allows you to make more material. That more material can be either just to replace what you've damaged or used up, or it can be for growth, and that, again, results in some level of evolution. All right, so if you're comparing this to a virus, right, what's the big one that viruses don't do?
They can't produce on their own.
Right. So the big thing is metabolism, right? There's, I will say there are a few exceptions in the microbial world of viruses that carry proteins that maybe have some function in metabolism of the larger organism. So it's usually they have metabolic genes that are co-opting some part of metabolism for another organism, but they're not in and of itself. They don't allow that organism without a host to generate energy, right? So they're really stealing a lot of resources from the host itself. So they have some components that allow them, they certainly have structure, right? They have some components that allow for growth, but they lack that ability in and of themselves to sort of garner energy and really be free living. Okay. In the microbial world, though, we'll see, especially when we talk about evolution, that boundary between free living is a little bit murky, right? So not always hard and fast truth, but in generally, these are properties that we would say all cells have. Of course, viruses also have evolution. Properties of some cells, we have things like differentiation. So the concept of differentiation, right, just has to do with having different cell states or cell life cycles, right? Being able to make sort of decisions about what you might do in one environment versus another environment. I think spore forming or endospore forming is a classic example of that, right? So you can respond to nutrient limitation or stress, and essentially differentiate yourself into a different form. A lot of cells communicate, meaning that they pass chemicals back and forth to each other, and they respond based on those chemicals, right? So essentially, it's like if I, tell you to do something, okay, you could choose to do it or not, right? But I'm giving you information that you then process and do something in response to that information, right? It's the same idea, but now we're using predominantly small molecules or small chemicals in the microbial world. A lot of cells are motile, or some cells are motile. And again, this is sort of a big one. a lot of them have mechanisms to essentially transfer large swaths of genetic information. And so when we classically talk about evolution, we generally mean vertical transmission of genetic information, right? Meaning, I have a child, I gave him 50% roughly of his genes, right? He got 50 from his dad. In the microbial world, usually it's close to 100%, right, from your parent. But a lot of them actually have mechanisms for getting big swaths of other information from the environment. And that's something we'll talk about. And certainly it confounds this sort of evolution part here. All right, so again, so there are major general differences between bacteria, archaea, and eukaryote. So then these again are going to be the microbial cell structures that we're going to focus on. I want to emphasize, we tend to loop bacteria and archaea together. Classically, these were called prokaryotes, right, which essentially means no nucleus. But I want to emphasize that evolutionarily, these organisms are as different from each other as they are from eukaryotes. And one of the things that you'll learn about in this class is that really eukaryotes are kind of chimeras of both, right? So they have a lot of information that is really very similar to what we see in bacteria, and much more similar to what we see in bacteria, compared to some features which are much more common or relevant to archaea. So in terms of structure though, right, they were classified based on organisms that didn't have a nucleus versus organisms that did. Even though structurally there's quite a few differences between bacteria and archaea. All right, so in general, when we're talking about sort of microbial eukaryotes, right, the key feature here is nucleus. A lot of microbial eukaryotes do have some form of cell wall, right, something to help sort of support that native structure. Not all of them do. You're probably familiar with things like amoeba, right, which don't have that great of a cell structure. So all that structure that they have is from what goes on internally to the cell. All right, so I mentioned this idea of prokaryotes or bacteria and archaea lack internal membranes. Gave an example at the very beginning. That's not really a universal truth, but it's largely true that almost every eukaryote has at least one internal structure, right? So even organisms that have lost mitochondria, right, still have a nucleus. Again, so there's some wiggle room there. And then in general, we think of size being an important characteristic here, an important factor, where you have, incredibly, generally very small bacteria and archaea versus large eukaryotic organisms. Sorry, I'm going to jump to this first, right? But there's a lot of overlap, right? And usually these are, we're going to call it somewhat exceptions to the rule. There are a couple of really gargantuan bacteria. We'll talk about this organism, Epilepisium. Some cyanobacteria are, also incredibly large. And there are pretty tiny eukaryotes. So there are a few microbial eukaryotes that are actually much more on a par with sizes of bacteria. And then you have some also incredibly large viruses. So viruses that look almost akin to cells. So they have these sort of gigantic viruses group that we're still kind of learning about, but this is an example of one, Pandora virus. So this is just to say that size is 90% of the time a good metric, right, to start to distinguish some of these groups. In general, viruses tend to be much smaller than cells. Generally, they're smaller than the cells they infect, but even that's not completely true. Pandora virus here is a virus of amoeba, which is still bigger than the Pandora virus, but much more comparable. It's why it's thought to look like a bacteria or be about the size of a bacteria. But yeah, so again, they're not always sort of these universal truths. So it's not size in particular is not something that you can use as sort of exclusionary. All right, so again, we're gonna focus predominantly on bacteria and archaea, but so most microbes lack a nucleus. And there are consequences to that. There are certainly some benefits, but why did a nucleus evolve? So essentially, you have better information storage. You have sort of more protection of your nucleic acids, right? If you have a nucleus, right? You have this added barrier for things, especially sort of selfish DNA elements or things that might, ultimately try to damage your genome, maybe chemical oxidizing agents, right? There are lots of things that you can kind of keep separate if you have a special compartment with sort of this extra barrier for your nucleic acid information. The trade-off here is that it's more work, right, to access that information. So potentially this is going to result in slower replication times, right? A little bit more energy to sort of transport some of this information long distances. The way that you can transfer information becomes a little bit different. So, horizontal gene transfer, because you have this barrier, because you're more protected, right, goes down. But that might also be part of the reason, right, eukaryotic organisms had to evolve other mechanisms for recombination. And so you have to have dedicated ways of getting new information into your cells. All right, and then another sort of challenge here, and one of the things that microbes are really able to do, and we'll talk about this more with transcription translation, is that you can really spatially couple processes if everything's kind of in one milieu. So things like transcription and translation are essentially coupled. You can start doing translation as soon as while transcription is happening. And this makes these organisms incredibly quick to respond to the environment. You can quickly shut things off. Whereas if you're a eukaryotic, there's a lot different sort of checks and balances to things like transcription and translation because you don't have that rapid sort of coupling process in place. And so regulation also becomes much more complicated. All right, so again, we talked about sizes and how this isn't necessarily a great discriminating factor, but this is just, looking at, in particular, across bacteria, right? There are several orders of magnitude differences in things like cell volume, size versus cell volume. And so this is, again, it makes it hard to kind of have sort of universal truths about sort of the ecology and the structure and function, but again, there's a lot of variation in these organisms. So hold on one second, because I think, all right, so, but what is the logic, or what is the benefit of being small? So why are the majority of microorganisms relatively small? Well, being small is actually much more energetically efficient. Shout out to short people. So essentially, if you're a lipid membrane, right, and some sort of vacuole, essentially, the space that you have to interact with your environment does not increase linearly with size, right? So essentially, the smaller you are, the more surface area you have to cover a particular amount of volume. So let's say you're an organism, right, that you're getting all of your energy, all of your information from sort of things going on the outside. I need to transport sugars in, right, to keep myself active. Having sort of this better surface area to volume ratio can be really important, right? So if you're a phototroph, you're making your own energy from light perhaps, this might be less of an issue, and we do see that a lot of the really large organisms in the microbial world tend to be things that are phototrophic or autotrophic. Sulfur oxidizers are another really big group of organisms. So this might be less of an issue, but especially when you're talking about boring old heterotrophs, right, like E. coli, even though they're in sort of a gut community, it's their ability, right, to access all of that energy really quickly that's really important. And so size is probably, or is almost definitely constrained, right, by that balance between how much energy it takes to maintain a cell, how large that cell can be, and how much access you can have to nutrients in your environment. Oh yeah, okay. All right, so I sort of flubbed this a little bit, so right, why does this matter for a microbe? But how do eukaryotics cheat this system? Anybody? Well, let's just do a quick discussion. So if you're a eukaryotic, right, how do you get around? Because obviously in general, right, cells are bigger. So how do you cheat this system if you're a eukaryotic? What do you guys come up with? Yeah.
Eukaryotes can actively transport nutrients across the membrane versus relying only on simple fusion.
Okay. Yeah, I mean, so microbes definitely also can do active transport. And yeah, I mean, there's potentially differences in terms of, active transport between the two. But as far as I know, a lot of the active transport systems between microbes and eukaryotes are actually relatively homologous. So a lot of the same transporter types get used. Any other guesses? And if I was a microbe, I would have stolen that from eukaryotes. So, any other guesses? Yeah.
Eukaryotes have organelles that create their energy.
Yeah. So eukaryotes have organelles, right? And organelles are sort of invaginations, right, of that surface area. And so that's actually a pretty big part of it, right? So if you are a eukaryote, how do you, the ability to sort of do some level of invagination, right? Have structures and surfaces within your cell to sort of organize within actually helps with that, increasing that surface area to volume ratio, right? Because you can now imagine that, your surface area, even though it's not exclusively accessing the outside, you can really sort of maximize interactions with a membrane, right, where a lot of, we're just gonna say, fun stuff happens, right? So that ability to have membrane structure is actually part of the cheat, right? It's part of the cheat with that surface area to volume ratio. It's not a perfect one, but certainly that's part of it. Anything else? All right, so that was a big one. Sorry, I think there's a PRS question next, and so I'm gonna skip it for next time. All right, and then I just want to sort of close with, all right, sometimes being big is better. Not always. But there are examples of organisms that have evolved traits like Thymargarita namibrensis, right? That essentially this organism has made sort of this incredibly large vacuole inside itself where it transports a lot. In this case, it's predominantly electron acceptor. So these organisms live in environments that things like oxygen fluctuate quite a bit. And so if it runs out of oxygen, it can utilize nitrate. And so this large vacuole in here is predominantly like highly concentrated nitrate that it can use if it runs out of oxygen or if it becomes an oxygen limited environment. And these little sort of refractile bodies here, these are elemental sulfur. So this organism is a sulfur oxidizer. So instead of using carbon, it uses these little sulfur granules. This is very analogous to fat. So these are really hydrophobic, kind of compact sulfur packets. And then it has this sort of large vacuole where it has its terminal electron acceptor. So this organism, right, kind of stores its own food, and this makes it incredibly resilient to very sort of energy dynamic environments, right? So sometimes there's a feast and sometimes there's a famine in the places that this organism lives. All right. So I'm gonna stop here, but next time we're gonna focus on talking about the cell envelope and everything that entails. Does anybody have any questions or comments? I'll be around for probably about